Hello, everyone. I'm Mark. I'm one of the co-founders at ProteinQure. So today I chose to talk about how we are scaling drug discovery to petascale on AWS cloud infrastructure. But before I talk about that specifically, I just want to give a high-level overview about ProteinQure in general. So ProteinQure is a biotechnology startup, and we're building a computational design platform for peptides, proteins, and antibodies. And our specialty especially is in the kind of small to large protein range. And we use natural as well as non-natural amino acids, which for the people that don't know what that means, for proteins, there's basically 20 natural building blocks that you can use to compose very complicated and intricate protein sequences. But there's actually hundreds of non-natural artificial building blocks that people have come up with and that you can use to kind of combine and generate proteins that haven't been discovered in nature and that are completely out of the range of what we've seen in existing data sets, which we think is really exciting because there's a lot of therapeutic potential there. Our expertise is in challenging and dynamic targets, for example, GPCR targets, which tend to have quite a lot of movement when they interact with drug molecules, which we can simulate on supercomputing infrastructure. And we have a strength in combining experimental data sets that we get from pharma companies, as well as machine learning and biophysical tools such as molecular dynamic simulations and docking simulations. So right now, we are a team of 13 employees in Toronto, and we're a very interdisciplinary team, and it's a mixture of structural and computational biologists, chemists, machine learning scientists, and software engineers. And we're essentially partnering with big pharma companies around certain diseases that they're interested in. And you can think of us as a molecules as a service company. We don't sell any of the software that we're developing. The only thing that we sell are the molecules that we have designed and engineered using our computational platform. So to dive a little bit deeper on this, basically one of the big problems when it comes to peptide and protein design specifically is that there's only very little data available, because traditionally pharma companies have mostly focused on what's called small molecules, which are roughly like 10 to 50 atoms large. And that's kind of like your typical over-the-counter drug tends to be a small molecule. Whereas in the protein and peptide world, there just hasn't been a lot of experimentation, which means that even though there's like 10 to the 65 possible sequences for relatively small proteins, we only have around 150,000 protein structures available to us today. So we have to kind of operate in a very low data environment. And we're also dealing with multi-property optimization, because kind of what we're trying to illustrate here on the left side, the pharma companies usually approach us with some form of initial hit, a molecule that they know kind of works on this particular disease, but it's just not like a drug molecule yet. It's not potent enough, or it lacks some sort of other property. And so when it comes to actually making a drug, there's all these aspects that you need to consider that go well beyond the potency of the molecule itself. For example, one thing is the molecule soluble in water? Like, will it actually kind of interact with your body in a proper way? Does the immune system have a bad reaction to this molecule? Does the molecule permeate cell membranes, which would be really important if you're trying to target a protein that is somewhere inside of a cell? So you need to really make sure that the delivery process is actually figured out, and that this drug molecule is stable enough to survive in your body long enough for it to actually get to the spot where it needs to be. So there's all these different properties that you kind of have to juggle at the same time, whilst also optimizing for the potency of the molecule itself. So that's why we've built out quite a lot of expertise in the area of multi-property optimization. And then there's this whole problem of unknown structure-activity relationships, where we just don't have good models that predict some of these drug-like properties, or even predict this potency. And that's why we're heavily banking on biophysical tools and simulation environments that help us kind of figure out and predict these properties in a better way. So kind of from a bird's eye perspective, our platform has sort of three pillars that all use kind of next generation methods. On the left side, we have anything relating to drug design, things like molecular docking simulations, which help you figure out if a drug molecule actually binds to the protein target that is linked to a certain disease. Sometimes we have to do protein folding. Depending on the length of these proteins, they start folding up in quite intricate three-dimensional shapes, as well as the multi-property optimization that I just mentioned. All of that is happening using our computing infrastructure, which is heavily cloud-based and uses a lot of existing tools that are out there for biotechnology and a lot of our own tools that we developed in-house. Then there's this whole pillar of drug validation, because it's one thing to design these molecules on a computer or in silico, as people like to call it, but we, at the end of the day, need to validate it. We often get the pharma partners to actually do all of the experiments for us, but sometimes we have to do experiments ourselves. For example, if a data set is too one-sided and our machine learning team comes back and says, okay, we need to do some more experiments to actually fill out the data set and get better generalization, we end up using our own wet lab infrastructure, as well as various CROs, contract research organizations, that can help with the synthesis and the conduction of experiments. And then on the right side, we have kind of the insights that we generate, because we believe that one of the big advantages of in silico methods is that you can actually start explaining why one molecule works and another one doesn't, because you actually have the simulations and the 3D structures, rather than just doing experiments in a lab and kind of just blindly testing molecules and doing local optimization around the ones that work. So in that case, that is heavily reliant on our data infrastructure, as well as all the machine learning models that we're building in-house. So before I dive into the cloud architecture and the reason why we scale to Petascale, I want to quickly explain why we actually need to get there in the first place. So this is kind of a very high-level, simplified view onto one of our workflows that we use on some of our projects, where you start out having an optimizer, for example, a genetic algorithm, or a human, defining a list of molecules which they want to test or screen virtually. So in this case, they might have a list of 100,000 proteins. They then submit them to our cloud workflow, which first runs some molecular simulation of some kind. So that could be a protein folding simulation or docking simulations. And after that has succeeded, we featureize the molecule. And then we train machine learning models or do inference using machine learning models to predict various properties. And out comes 100,000 predictions, and the optimizer or human then decides that they might want to test another 100,000 or another 500,000 proteins. And so we keep iterating through the cycle until we converge on a list of promising molecules, which we think have desirable properties, which we then can submit to our pharma partners. So because we have to deal with very large numbers of proteins, so a typical project, we test around 1 million proteins in silico, we need to be able to really scale this up to a very large size in order to be able to actually achieve that kind of throughput. And so I want to quickly talk about how we started doing this. Basically, at the beginning, this was one particular project that we worked on. We had this like pretty convoluted pipeline that was happening. And the problem was that it wasn't really a pipeline. It was more of a workflow, in the sense that it actually had to be done manually. There was a lot of hand-holding involved. And you can kind of think about it as like a fire bucket brigade, where you would run a step and then you take the outputs from that step and you manually kind of walk over to the next step and run that one. And it just ended up basically one of our developers would sit there with 4 terminals open, kind of monitoring the files coming in on the shared storage, overlooking the computations, the logs, and kind of trying to manage all of this in parallel. And that was obviously not ideal and also not very scalable. And so at the time when we were running this, our performance was roughly at like 5,000 concurrent Kubernetes pods and 6,000 CPU cores. And we decided that this is not very scalable and we need to change things. And so what we did during the lifetime of this customer project is that we went from these 5,000 pods and 6,000 CPU cores to 37,000 running pods and 31,000 CPU cores. And I want to talk about kind of how we got there, what technologies we used, and what pitfalls we encountered on that way. So the first thing was that we realized, okay, we got to move away from our internal tools that we developed, which we called Dart and Gust, and we decided to move to a open source tool called Argo. So what is Argo? Argo Workflows is an open source container native workflow engine, which helps you to orchestrate a lot of parallel jobs on a Kubernetes cluster. So you can kind of think about it as defining a workflow, and each step in the workflow is a Docker container. And it's cloud agnostic in the sense that you could take this and run it on any kind of cluster. You could run it on Google Cloud, AWS, whatnot. Wherever you can spin up a Kubernetes cluster, you can run Argo. And the nice thing is that it really puts a cloud scale supercomputer at your fingertips, and you can start composing these very intricate, very elaborate workflows. And Argo kind of handles a lot of this for you and kind of moves data between the various steps and really makes that easy. And then you can just scale it up and run hundreds of these workflows or thousands of these workflows in parallel. So to show a little bit of code, Argo uses YAML. And one example of how you can define a workflow step using Argo is just by writing a couple lines like this. In this case, the step is called generate random integer. It's a script step, and you define which Docker image the step is supposed to be running in. So in this case, we're running Python 3.6. We're running the Python command, and then you can literally just write down the source code that you want to run in this particular step. So here we're just generating a random integer and printing it out. And then once you start building these individual modules or these steps, you can start chaining them together. So in this case, I have a dummy workflow here called protein design, and it consists of 4 different steps. Step 1 is preparing some data, and so that's referring to some step template similar to the one that I just showed you that is defined elsewhere. Step 2 is simulating the protein, and step 2b is actually running in parallel with step 2a, and it's uploading the data that we prepared in step 1. And then step 3 is featurizing the protein, and you can extend that as much as you want. And what you get out of that is these very intricate workflows that kind of branch off and come back together. And all of the data that is flowing through this workflow is kind of handled by the workflow itself. And you get really nice log output. And you could now, this is branching out into 3 branches, but it could theoretically branch out into thousands of branches. How do you run that at the end of the day? You have this workflow YAML file, and you just run Argo submit if you have the Argo CLI installed and Argo deployed on your Kubernetes cluster. And it's also parameterizable, so you can pass a parameter file. So in the case, for example, in our case, we have different projects that use different protein targets, different hyperparameters, and so on. All of these things you can pass with this parameters file. So far, so good. So we thought, let's get started. Let's rewrite that old bucket brigade workflow as an Argo workflow. And we did it, and it worked. But the problem was it was still kind of suboptimal. Like our performance didn't really improve. We had like lots of dead ends in the workflows, and there's just a lot of problems. So we kind of iterated on it, started doing incremental improvements, and eventually we scaled it up to like hundreds of proteins, and we started running into problems. Mainly, like the main container in a Kubernetes pod would finish its work, but the wait container would keep running for minutes. So don't worry if you don't understand what that means. I'm quickly going to give you an overview what a Kubernetes pod looks like when you're running Argo. Basically, a pod consists of 3 Docker containers. There's the main Docker container, which is actually running the computation. So for example, a molecular simulation. And then there's these 2 sidecar containers. The init container runs at the beginning and downloads all the data, for example, from Amazon S3 or some network file storage, and gets all the data ready for the main container to consume. And the wait container runs after the main container has finished, and it uploads all the data that was generated as part of that molecular simulation, for example, and uploads it to S3 and NFS. And for some reason, like this wait container just kept running for ages, even though the computation itself was done. So we kind of started looking into that using Grafana, which we use to monitor our cluster. And we started wondering, like, is it the CPU? Is it running out of memory? Are we struggling with disk I/O? But it wasn't the case. And what really was the problem was that there was just a lot of network traffic. And so what we started realizing is that we would run this in like parallel on many, many proteins. At that point, we were running like roughly 1,000 proteins in parallel. And they would all access that same file storage and would all try to connect to it and send data around and stuff like that. And that just started jamming up the entire cluster. So in our case, the solution was to just throw more money at it at first, just to get the computations going. And then ultimately, we removed all the dependencies on this network file storage and decided to move to S3 directly. And that kind of already boosted our performance slightly, and we went up to like 9,000 CPU cores. But the next problem then was that we actually ended up getting our CPUs throttled, because all these simulation pods and machine learning pods that would run in parallel would all kind of battle and fight for the CPU that was available, which you can kind of think of as like a crabs in a bucket problem. Whereas if you have a lot of crabs in a bucket, they all just like kind of pull at each other. So like the crab that is trying to crawl out of it, it keeps getting pulled in. So none of the jobs really get finished. They all just get like a couple milliseconds on the CPU, but are all kind of jamming and throttling each other. So in this case, we had to adjust resource limits, and Argo allows you to kind of define how much CPU and memory you want to allocate for all these different individual steps. And that, again, boosted our performance slightly. But every time we thought, okay, great, we solved it. We can now scale it up to like 1 million molecules. But then again, we ran into another problem. In this case, we were running into an issue with S3, because S3 is limiting how many files you can upload at any given point to 3,500 uploads per second. And we were hitting that number and exceeding it by far, because each protein molecule was generating 180 files, which we all needed to save. And we just never really encountered this problem before, because it was masked by the NFS issue that was like slowing us down even more, that we solved beforehand. So in that case, our solution was to use artifact archiving, which is Argo kind of allowing you to zip the files up before uploading them. Sounds simple, but at the end of the day, it like really was a big bottleneck for us. And so we actually, for the first time, like achieved like a teraflop compute scale on AWS with like roughly 18,000 cores. So it was like the first time that we actually thought, okay, we're getting somewhere. But then ultimately, we ran into the biggest problem, because it had to do with Argo. Argo is an open source project. It comes out of Intuit, but they don't really have much developers working on it. And one of the problems is that usually if you run a workflow, you want to see these tick boxes on all the different tasks. But in our case, we just ended up workflows kind of ticking off a box, and then just nothing happened afterwards. They wouldn't spawn the follow-up tasks. And so we reached out to the lead maintainer, Alex, on GitHub, and I want to give him a shout out, because his response time was amazing. And we ended up solving the problem, or he ended up solving the problem, by pushing an engineering build on that same day, which I think is a great example for how open source, and also contributing back to it, which we've been doing in the past weeks quite a lot, is just working at the end of the day. And ultimately, using that engineering build, we were able to scale this computation to 1 million proteins in parallel. We hit 1.3 petaflops per second on AWS with 31,000 cores, which I think is just amazing that that's possible using essentially a combination of open source code and publicly available cloud infrastructure. And to put things into perspective, it essentially took our team of 4 engineers and a lot of existing software, as well as all the software that we developed in-house, to essentially get the equivalent of a little supercomputer. So if you look at the list of top 500 supercomputers in 2020, the number 500 has 1.3 petaflops. So it's kind of cool that this is all possible without actually buying a whole cupboard full of compute resources. So to wrap it up, I wanted to touch like 3 little tips just from kind of the live ProteinQure that we've been recently encountering. Tip number 1 is try a design sprint. It's an amazing little method that was developed by some folks at Google Ventures. And the idea is that you kind of compress product development into a single week. You kind of like trying to just brainstorm and quickly create some prototypes of new pieces of code, I guess in our case, but it could be anything, like build new tools that could be useful in your company, and then immediately test them with your customers or users. In our case, that was our in-house scientists. And doing that in a week and just really sitting down and focusing on that helped us a lot, coming up with a lot of new tools that we're currently developing to boost our scientists' performance. The other thing is try out GatherTown, which is a great tool to make your work from home environment a little bit better. You essentially have this Game Boy style looking environment where you can build your own office, and you kind of get more random encounters with your coworkers. You can walk over to someone's desk and ask them a question because their video and audio fades in and fades out. And it really has helped us a lot to kind of connect the team a little bit more than what you can do with Slack and Zoom.
Somebody has asked, some of your technologies can be applied to small molecules as well. Is this being explored?
No. I guess we founded this company because we believe that proteins, peptides, antibodies are the future of drug discovery, and we exclusively focus on that. And also kind of all of our hiring and our entire team is very trained, and most of them come out of academia, and they focus on protein design and protein simulations. So no, we don't focus on small molecules.
Where do you expect to go next? Future plans or discoveries?
Wow, very open-ended. I guess where we're planning to go next, maybe just with the cloud stuff that I was showing. 1 petaflop is just the beginning for us. We want to scale these things up way more and really build a modular platform where we can kind of encourage. The problem is for us, the biologists, even though they're computational biologists, they're not software engineers, and we want to kind of lower that barrier for a biologist to be able to develop their own very intricate workflows and kind of protein design loops that you can maybe close with an optimization algorithm and so on. And so we're really trying to currently develop a lot of tools that make that easier for them, make it more accessible, such that you don't need a bunch of software engineers that are constantly kind of working together with them in order to make these things work.
Given the 10 plus atoms in a typical amino acid, can you use the Born-Oppenheimer approximation? Your simulations of molecular dynamics? If not, what do you use?
Wow, it's a very good question. Unfortunately, I don't know the answer for it because I'm not a computational biologist who writes protein simulation code. So I don't know, unfortunately. My background is in quantum physics, and one of the issues, obviously, with these protein molecules is that you cannot do any quantum simulations of these molecules because they're way too large. So typically, with molecular dynamics simulations, you approach these simulations by essentially saying, okay, we approximate an atom just as a point in space, and we're not modeling all of its electrons and all of these kind of intricacies that would be there, and you're just essentially simulating these proteins using classical physics, Newtonian physics, which makes the simulation easier, but also slightly lower resolution. But that's kind of the price you pay for that.
What are the challenges in working in this field? Is data quality and availability a problem, and how do you improve them?
Yeah, I think that is, you kind of answered the question yourself. I would say data availability is a huge problem. The fact that we're dealing with this low data environment just makes it really hard for us to develop any kind of good machine learning models. I mean, it's possible, but it's just really challenging every single time because you don't have terabytes of data that you can train your very deep neural nets on. But you got to really kind of dive into the statistics, and sometimes we actually have to go out and do more experiments, make sure that our data sets are more balanced, and that's the really tricky part. The fact that every year we only get a couple hundred more structures because it just takes so much time for people to actually get the crystal structure of a protein. And the simulations at the same time are also extremely slow. So we're kind of working on a lot of techniques that also help you circumvent the simulations part. And kind of there's various types of ways of building ML models that kind of help you approximate protein folding predictions as well as docking and so on. So I think really getting more data in that field, more experimentation and more public data sets is a big challenge and would help greatly. Not just us, but the entire field, even academia.
There are multiple sources of protein denaturation.
Mm-hmm.
pH, heat, heavy metals, and others. Do you take those into account into your work?
Yes. I mean, at the end of the day, all of these are kind of different properties. It always depends on our pharma partners. They come in and they have usually some properties in mind which they want to focus on, and some which they are okay not to focus on because maybe they have like in some later stages in their pipeline, they have ways of optimizing the molecules, for example, to like not have them denatured by heavy metals and stuff like that. But we do focus on kind of the main pieces that lead to protein denaturation. And in our multi-property optimization, try to make sure that the molecules we deliver already kind of check a lot of the boxes, because ultimately that it's not really worth it to have a great potent molecule that is not soluble in water, for example. So I think that's one of the reasons why I guess like pharma companies like to work with us, is that we factor these things in from the very beginning when we design these molecules.
Do your clients ask you to evaluate the metabolism and pharmacokinetics and toxicology evaluation for the leads that you generate? Asking as a biologist.
They do not ask us that because, like, if you kind of think about the drug discovery pipeline, we're at the very, very early stages, before any toxicology, before any animal studies, before any clinical trials. So we're kind of mainly responsible for giving them the first lead compounds, the first hits. They usually come to us, and on some of these projects, they've tried and worked on them for several years, had experimental teams working on it, and they just hit a wall. And that's when they approach us and say, okay, hey, can you try out these computational techniques? Can you try and find some molecules that work that we can start putting into our pipeline? So usually these things are being considered later on, and they require quite a lot of experiments and stuff like that. So no, we don't usually are asked to focus on those.